In this video, we are going to see what is microbial nutrient medium. Microbial nutrient medium is a mixture of different chemicals which provides all the nutrients for the growth of microorganisms in required amount. It should provide all the basic elements required for the growth and minerals for the growth of those microorganisms that you want to cultivate. The medium should have suitable carbon source, nitrogen source. Organic molecules always contain carbon, hydrogen and oxygen. Many of those organic molecules they are interconvertible. That means if you provide carbohydrates in the medium, the majority of the microorganisms they can convert these carbohydrates into lipids. So there is no need to add separate lipids in the microbial media. Similarly, vitamins can be synthesized by microorganisms only if the microorganisms are not having those pathways for the synthesis of vitamins. Those need to be added in the medium separately. So your medium should have all the basic element sources plus different types of salts or minerals which are required, plus it should have salts which are going to maintain the osmotic balance in the medium. Sometime the medium need to be have required to have particular pH. So pH can be adjusted by different types of molecules. So we'll see what are the factors which decides the composition of the medium. First you need to have idea about the cell composition, because the cells when they are growing they are going to obtain nutrients from the medium. So your medium should provide all the required elements so that these biomolecules which are forming the cells can be synthesized. So composition of the cell gives you idea about the components or chemicals which need to be present in the medium. The second factor which decides the composition of the medium is metabolic mode of that microorganisms. If the microorganism is autotrophic, then carbon can be provided in the form of CO2 which can be used from the atmosphere. If the organisms are heterotrophic, then carbon should be present in organic form. If you want to cultivate chemolithotrophs, the elements from reactions of which the energy is obtained, those elements should be present in your medium. The genetic makeup of that organism need to be considered while designing the medium. Some cultures can utilize some sugars and are not able to utilize some types of sugars or nutrients. So if you have idea about this type of mechanism or this type of ability of the culture, then suitable components or nutrients can be added in the medium. If you want to use that medium for just growth of the organisms, then you can have crude components in that medium. And if you want to find out effect of particular nutrients on the growth of organism, it is always better to have a defined medium where you are using the pure components and the amounts of those pure components is also known. The duration for which you want to support the microbial growth is also important while designing a medium. If you want to just have a growth of microorganisms for a short period of time, then limited amount of nutrients may be added in the medium. But if you want to use that medium for fermentation for longer duration like 10 to 15 days, you need to see that sufficient amount of nutrients are provided in the medium. Some of the nutrients they are required in more quantity, and some of the nutrients like vitamins or minerals they are required in less quantity. So depending upon their requirements, these components should be added in the medium. Downstream processing that you will be doing for purification of product from the medium is also important. If your product can be easily separated, then you can use crude components in the medium. And if your product is difficult to purify from the fermentation medium, it is always better to use pure components or defined medium for cultivation of those microorganisms. The media is available into two forms: solid medium, where you use some type of solidifying agents like agar, and the other form of medium which is available is broth, which is a liquid medium. So if you want to just enrich the culture, then in that case you can go for the liquid medium. But if you want to isolate the colonies from a mixed culture or from natural samples, it is always better to spread those natural samples with required dilutions on the plates where the cells get localized at particular points and they can form isolated colonies. On the basis of colony morphologies, you can try to identify the cultures and purify the cultures. So, in short, microbiological nutrient medium is like a balanced diet which provides all the nutrients required for growth of those microorganisms. So, microbiological nutrient medium are agar or broth-based substances that provides essential nutrients for growth, cultivation, and maintenance of microorganisms in the laboratory settings. So, you always try to mimic the natural conditions where the organisms are present, and you always try to provide the nutrients so that those organisms can be grown in the medium. There are certain pathogens which multiply in blood. So, and that's why they require specific components from the blood for their growth. So organisms which have such specific needs for the growth, they are called as fastidious organism, and you need to take care of their requirements. You need to add some of these components in the medium so that these organisms can properly grow in the medium. These media they contains carbohydrates, proteins, salts, and other necessary elements to support the microbial growth. As mentioned earlier, some of these components they are interconvertible, but you need to see that all the required elements they are present in the medium. The salt should be present to maintain the osmotic pressure, and the pH should be adjusted, which should be suitable for the growth of that microorganism. Nutrient mediums you can tailor or design for specific type of microorganisms, or it can be used for general purpose. So media they are very much important in microbiological laboratories because they help in isolation of cultures from natural sources where always you get mixture of cultures or mixed cultures are present. Second thing, it helps in cultivation of microorganisms, which is always required to study the properties of that microorganisms. There are certain media which helps in identification of the cultures. For example, differential medium where you can find out some metabolic properties of the cultures. These types of media helps in diagnosis and detection of different types of pathogens in microbiological pathology laboratories. There are certain media which are selective medium on which only particular type or particular group of organisms can be grown. So this type of media helps in growing only particular type of pathogens or organisms from natural samples. Media can be used for biochemical test where you can use different types of carbon sources or other components. Some indicator dyes can be added and you can find out what is the metabolic property of that culture. This helps in identification of the culture. So in short, the media helps in isolation of cultures, cultivation of microorganisms, identification of microorganisms, and in diagnosis or medicine also. Thank you all.